Assalamualaikum warahmatullahi wabarakatuh, and a very good afternoon to Dr. Farah and to all our fellow friends. So our group will be covering on the microbiology laboratory, and this is our presentation on the microbiology laboratory design. And these are our group members. Okay, let us start with the purpose of microbiology lab. As well known, microbes are infectious agents that are common in nature but cannot be seen with the naked eye. And some microbes can cause diseases to human. As well known, microbes are viruses, fungus, bacteria, protozoa, and also microscopic parasites. And the two major functions of infection diagnosis in microbe labs are clinical and epidemiological. Some infections, such as the acute meningitis, sepsis, or severe pneumonia, they require quick clinical diagnosis and empirical treatment without identification of the causative species or assessment of antibiotic susceptibility. But lab testing is useful and it is utilized to confirm the diagnosis when there is a clinical suspicion of infection. Moreover, the majority of healthcare associated infections, HAI, are caused by bacteria and fungus that could be more resistant to antibiotics than community acquired pathogens. Sometimes their susceptibility to antibiotics is unpredictable. Hence, etiological HAI diagnosis is essential for targeted antimicrobial therapy. This kind of therapy can eradicate the pathogens sooner, prevent the transmission of disease, enhance the patient's condition, and thus improve the whole situation. And on the other hand, the microbiology lab also plays a role in effective infection prevention and control (IPC), especially in the control of HAI, such as through outbreak management, performing additional tests for epidemiologic studies, bacterial and fungal typing, HAI surveillance, and reports about new alert microbes or unusual resistance. These are several diagnostic tests that are available in microbiology lab. As you can see, the bacteriology tests, most of it involve culture, and the samples required are CSF, blood, urine, sterile body fluids, nasal swab, throat swab, sputum, pus, stool, and there's a lot more like vaginal swab, ear swab, eye swab, and so on. Depends on the disease. And the next one is mycobacteriology test, which involve the sputum for EFB detection and TB test, and also the serology test. Serology test, of course, it involves blood, which can be used to detect dengue, hepatitis, HIV, toxoplasma, mycoplasma, and so on.
Proceed with segregation of areas. Here is full floor plan for microbiology lab. First is receiving and processing room where all specimens are received, accessioned and divided. Then sample preparation room where having a culture room to culture bacteria and urinalysis room for urine testing. And this is microbiology lab where bacteria identification test, bacteria growth observation and bacteria susceptibility was done. Then proceed with lab equipment. There are many basic and important equipment used in microbe lab such as balancer, microscope, mixer, autoclave and incubator. Here are also some of lab equipments are used and crucial such as laminar flow, fridge, sonicator, homogenizer, pH water. And then we move on to automated equipment. First is total laboratory automated system, such as BD Kiastra. This equipment is two-way conveyor linked to all working sites in microbe lab. Second is cell colony counter. Electronic colony counter is used to calculate bacteria colony form on plate. The electronic cell counter is used to calculate bacteria cell in liquid sample. Third is mass spectrophotometer such as MALDI TOF. This is used for microbe identification and very rapid and accurate. Finally is susceptibility machine such as VTEC2 Compact, where it performs antibiotic susceptibility test on bacteria. Other than laboratory equipments, there are also laboratory apparatus such as beaker, volumetry flask, petri dish, pipettes, and etc. All these laboratory equipments and laboratory apparatus is very important and usually used in lab.
Reagents and chemicals that will be used in microbiology lab. Firstly, stains. Stains include routine stains, which is gram stain. Its solutions is crystal violet and iodine. Special stains include carboxyl, silver nitrate, India ink, and acridine orange. Counter stains are safranin and methylene blue. Decolorizers are alcohol and acid alcohol. Culture media. Culture media includes selective media and differential media, and also media that will be used in biochemical reactions. Those are examples for the most important media that will be used in microbiology lab. Reagents also include antimicrobial sensitivity tests, which are paper discs containing the antibiotic and culture media, antimicrobial agents as a solution, and prose media. Serological test reagents include kits for CRP test, ASO, rheumatoid factor, RPR, SLE, and ELISA. Reagents also include disinfectants, which are alcohol, chlorine-releasing agents, phenol, and quaternate ammonium compounds. Lab ventilation. Lab ventilation is important because it protects the worker from inhalation of hazardous chemicals and infectious microorganisms, protects the working environment from being contaminated, and also protects the external environment by filtering the air exhausted from the lab. General ventilation system. It's a general system of air ducts located throughout the space of the laboratory. There are two types of ducts, supply ducts and exhausted ducts. Supply ducts through which the supply air is filtered and enters the lab, while exhaust ducts through which the lab exhaust air is filtered using carbon high efficiency particulate air filter, then processed by centrifugal fan and safely propelled outside the building. Supply and exhaust ducts will be properly separated to avoid mixing of polluted air with clean air. Also, laminar flow chamber will play a role in lab ventilation. It's a glass-fitted chamber, blows air from the surrounding and passes it through a high-efficiency particulate air filter to make it dust and microbe free. Then the air passes through the chamber and comes out through the open front door to prevent the outside air from entering into the chamber. Also, there an UV lamp fitted inside the chamber to sterilize the chamber before operation. Since laminar flow chamber blow air back to the area of the worker, it cannot be used in the infectious agents or to handle the infectious agents, but it can be used for bacterial culture experiment, preparing culture media, and assembling of sterile equipment. Biosafety cabinets. It's quite similar to the lamina, but it differs from it by cleaning the particulates away out of the lab air and remove the airborne contaminant inside the hood, so it can be used to deal with the potentially infectious agents.
Now let's look into equipment management. First, all equipments must be recorded in the inventory record. It should also be calibrated prior use. Performance verification should be done to verify manufacturer's claim on the equipment's performance. Operating procedure also should be established for each equipment, followed by maintenance program to ensure good performance of equipment. Operator training on troubleshoot is very important to ensure a smooth working process. These are several documents that should be established for equipment management. They are inventory record, calibration schedule, operating procedure, maintenance logbook, and usage logbook. Centralized computed inventory system would be the best to be adopted since it provides easy access on equipment's information. Equipment labeling is important. Equipment location plan can be put inside the room to help one navigate equipment's location. Calibration schedule provide guidelines on method and frequency of an equipment calibration. Equipment specification explain the equipment details, and operating procedure provide step-by-step guide on operating the machine. Usage logbook, maintenance schedule, and maintenance logbook should also be provided for each equipment. Several processes in chemical management are order and receive, inventory, labeling, storage, and disposal. Chemical inventory can be integrated with the main inventory system for easy tracking. MSDS should be a guidance for storage of chemical. SOP document should also be provided for each chemical, and labeling should not be removed if chemical is still in use. Chemicals can be stored in cabinet that suits its characteristic, and storage plan can be prepared to navigate operator to the chemical. Waste area should be provided in the room, and waste record should be prepared to track the chemical stock.
Now, let's look into the Laboratory Management System, or also known as LIMS. Lab Information System is a software that is developed to support laboratory operations. It can record, manage, and store data for clinical laboratories, and traditionally it is adapted to send the lab test orders directly to lab instruments, tracking those orders and then recording the results, usually to a searchable database. LIMS serves the main purpose of enhancing the efficiency in laboratory functioning by cutting down the manual task. There are a few stages in LIMS. There are 5 core stages. Receiving and logging in of samples is the primary step. Then it is followed by sample assign, schedule, and further tracking before being analyzed, sample processing and quality check, data storage with sample analysis, and final report generation after inspection, approval, and compilation of sample data. LIMS is pretty time saving, and it can be used to automate workflows, and for this reason, the system should be used to automate record keeping. When existing procedures and methods in LIMS are codified, it delegates decision making to the software. Like, it will be able to automatically allot work to the scientists as well as suggest instruments based on preset rules. This way, the system won't look up what staff requires to do with the sample and where it will need to go next, as the LMS automatically provides this info. The advantages of having laboratory management system are workflow automation, meet regulatory compliance, improve data quality, real-time tracking and time saving, interface with instruments, and improve turnaround times. Next, let's look into waste management. The most important step in the disposal of waste is the correct identification of the waste type and determining whether it needs to be treated prior to disposal. First step is identifying the waste type. Identifying the waste type in waste management will make it easier to segregate and dispose the waste. The type of waste that can be obtained from microbiological lab waste are human anatomical waste, human cell lines, human biohazard group 1, human biohazard group 2, human biohazard group 3, genetically modified organisms, specified animal pathogen order organisms, non-human cell lines, soft plastic pipettes, hard plastic pipettes, tissue culture plastics, electrophoresis or western blotting waste, and toxins. Next, appropriate destruction method. Destruction method can be divided into 2 steps, mainly autoclaving at 134 degrees Celsius and validated chemical destruction using Virkon, bleach, or other chemicals. The moist heat from autoclaving destroys microorganisms by the irreversible coagulation and denaturation of enzymes and structural proteins. But moist heat will not destroy endotoxin, and by the nature of destroying gram-negative bacteria can actually contribute to increased endotoxin levels through cell lysis. Hence, chemical destructions are used in this situation. Moving on to laboratory biological waste disposal guidance. Prior to disposal, the waste has to be segregated to non-sharp waste and sharp waste. This is to reduce the chance of injury or needle pricking while disposal of rubbish. Examples of non-sharp waste are untreated infectious waste containing chemicals, untreated infectious waste not containing chemicals, cytotoxic or cytostatic waste, and anatomical waste. Example of sharp waste are sharp hard plastic pipette waste, sharp cytotoxic or cytostatic, sharp used to dissect anatomical material, and sharp boxes. These wastes are then further discarded into the specific colored bins. These bins have several colors. The first is red lidded box that is available for anatomical waste, followed by purple lidded boxes
that are available for cytotoxic or cytostatic waste, yellow-lidded boxes that are available for pathogenic or GMO contaminated waste or certain chemicals, orange-lined biobins that are available for untreated pathogenic or GMO contaminated waste, and finally, tiger stripe-lined biobins that are available for offensive waste or treated pathogenic or GMO contaminated waste.
Recruitment of microbiology lab technician. The requirements are: they must have a microbiology background or other related fields, have working experience in the microbe lab, proficient in standard lab equipment and techniques, familiarity with standard lab safety procedures, and attention to detail skill. This is important because they are working with very small organisms and materials that can easily be contaminated or cause errors in the results. Being able to perform tasks with precision ensure that the results of experiments are accurate and that the samples are not contaminated. The intent of training is to enhance staff hands-on skills and performance, helps in handling real-time challenges and decision-making, and to provide the staff with the knowledge and skills necessary to be competent in their assigned duties and responsibilities. New staff need orientation and training. Every facility is different, and regardless of how much lab experience an individual has, new staff must be trained in the following areas, which are their assigned duties and responsibilities, lab policies, lab manuals for all of the tests that they will perform, lab safety practice, and computer system for recording and reporting. On the other hand, current staff need continuous training when new process and test procedures are implemented, when current process and test procedures are changed, and whenever the need for additional training is identified. For example, failing the competency assessment. Competency assessment. It is the means to confirm that training is effective and that the staff are capable of performing assigned tasks according to the defined procedure, and can fulfill their duties and responsibilities and get accurate lab results. Competency assessment should be conducted every 6 months during the first year of employment for new staff, once a year for all staff, and before the introduction of any new strategies, procedures, or instrumentation. Competency assessment is an opportunity to provide continuing education and performance feedback to all staff.
One of the important aspects in designing lab includes guideline and quality. Guidelines are needed so that good laboratory designs allow scientists as well as technologists to conduct their work in the most efficient and safe manner and in a laboratory that will be in compliance with all relevant regulations, while quality management assures the reliability of all aspects of the operation. Regulations that we can follow in designing microbiology lab are guidelines from College of Pathologists, Academy of Medicine Malaysia. We also have guidelines for working with infectious and potentially infectious agent/materials and microbial toxin under Malaysia Laboratory Biosafety and Biosecurity Policy and Guideline. We also adopt safety and health aspect in laboratory from OSHA standard and the Occupational Safety and Health Act, United States. As in Malaysia, OSHA guidelines are not directly related to clinical microbiology lab. Malaysia, regulation related to pathology laboratory is the Pathology Laboratory Act 2007. This act allows laboratory to be accountable to the public, meets required standards of practice, participates in quality assurance programs, run by qualified staff, comply with safety requirements, and also perform continuous audit. However, this act have yet to be implemented strictly. Hence, we can go for other quality management system such as ISO 15189. ISO 15189 certification is an international standard that specifies the quality management system requirement particular to medical laboratory. Next, we have ISO 17025, and this certification enables our laboratory to demonstrate that we can operate competently and generate valid results, thereby promoting confidence in our work both nationally and around the world. We can also go for ISO 9001, which is the international standard that specifies requirements for a quality management system QMS. By using this standard, we can demonstrate the ability to consistently provide products and services that meet customer and regulatory requirements. Finally, our lab will also have the internal quality assessment as well as external quality assessment program.